the quiz tomorrow. But what we will do is we will finish up just about all the material for our exam, so that's good. It will be pretty much wrapped up like 2 weeks in advance. Exam 2 starts with that disinfectants lecture with the heat and autoclave and all that. And then it kind of goes through this chapter, which is all about the pathogenesis. So we started the pathogenesis, and we're going to finish up most of it today, actually, or just about all of it today. But we introduced it on Monday, and I kind of had that flow chart with like the boxes. And I said we're going to basically kind of focus on the middle of that pathway, which is how microbes basically kind of slip into our system and avoid our immune system, and then how they actually cause damage to our bodies as well. So a big component of what we're going to talk about today are those toxins, because we've kind of briefly mentioned toxins in chapter 4 when we talked about plasmids, that plasmids can carry information for toxins. So today we're going to kind of delve into the toxins also. We left off here though because basically we established on Monday, we said the first big step is attachment. And attachment is huge for especially the viruses, right? Because viruses are obligate intracellular parasites, but bacteria also like to attach as well. Because if they attach somewhere in your body, then they'll have their little niche. And then a lot of times they can create what we call biofilms, right? Bacteria, though, we started to list kind of like a variety of extracellular enzymes that bacteria can release out. And these enzymes can have different functions for the organisms. I'm only giving you kind of some examples. I notice your textbook also has additional examples, but we mentioned coagulase, kinases, collagenase was another one. Hyaluronidase is kind of a interesting enzyme, but this is an enzyme that breaks down a type of sugar that you have in between your cells. It kind of glues your cells together. So hyaluronidase is kind of like a good way of sealing your cells kind of like together. So that's why it says it's a sugar, and you find it a lot of times in connective tissues. So bacteria can release an enzyme that breaks that down. It's kind of the same idea as collagenase, and they'll use that to their advantage. It can kind of spread, they can get deeper into your tissues and kind of hide out. So it's a big advantage if you can make these extracellular enzymes. And we alluded to this on Monday too, that flesh-eating bacteria. We're going to talk about that later on in the semester. But a lot of these really aggressive type of organisms that can cause a lot of damage are kind of notorious for making these extracellular enzymes. So the flesh-eating Streptococcus pyogenes, that organism is kind of like notorious for making hyaluronidase and collagenase, and a lot of these we call them invasive enzymes. All right. So our body, in our bodies we have tissues, we have connective tissues that help to form barriers, of course. But organisms find a way to break down these barriers, unfortunately. And they can still slip through in our body. So this picture is kind of like an up-close view of those enzymes and what they're doing. In lab, we talked about coagulase already. That if they make coagulase, they can kind of form like blood clots around themselves, which is a big advantage because then they can kind of shield themselves off, and your immune system doesn't see the bacteria. Or they can make a kinase. We said kinases do the opposite. Remember, they break down the blood clots? So they break it down, and then they can go ahead and spread throughout your body. And then on top, collagenase and hyaluronidase, these are both enzymes that affect either collagen, which is more like a protein that you find in your connective tissue, or hyaluronic acid. And hyaluronic acid is actually sugar, though. So this is a sugar that you also find in your tissues, and by breaking down either the sugars or the protein kind of support network underneath, either way, it allows organisms to kind of slip through and hide out deep into your tissues. So that's why it says bacteria are invading deeper in your tissues, which is good for them, not so good for us. So you guys actually already know about this test, because remember we briefly mentioned it in lab, that coagulase test. So there are some experiments you can do, where you can actually test for the presence of a particular extracellular enzyme like coagulase. And the coagulase test is really easy to do. It's just kind of the downside is it's pretty expensive because you need that plasma. So, and usually rabbit plasma is kind of the easiest thing to do, but it's kind of an expensive test. But you can just inoculate it. And since we said Staph aureus was like the one that makes coagulase, you can differentiate between Staph aureus from all the other types of staph, which are all negative for coagulase. And if you inoculated Staph aureus into the rabbit plasma, that plasma would actually gel up. So it would start clotting. And when you pull it out of the incubator, it would kind of be like the normal gelatin tall, like nothing would move. But in negative result, it would remain fluid. So, but what's kind of interesting is we found a way though to commercialize some of these extracellular enzymes. We mentioned the kinases, since kinases break down blood clots. They've actually found a way to commercialize the kinases, and so they have them here in vials as a way of breaking down clots in us. And so we kind of use that to our advantage. So somebody who might have a blockage in a blood vessel, that might put them at risk for heart damage or heart attack, you can actually give them intravenously a kinase enzyme to help break down that clot for them. So it's kind of nice. It's a good advantage, good application of the extracellular enzyme, the kinases. I only have one more extracellular enzyme, and that's the IgA protease. That's the last one. So we've mentioned two that are related to blood clots, and two that are more related to breaking down either proteins or sugars in our connective tissues. Protease is a little bit different. It says IgA, and we haven't talked about antibodies yet, so IgA doesn't really mean anything to us yet, but IgA is a type of antibody that we have in our bodies. And what's really nice about IgA is the antibody that you find on mucosal surfaces. So we'll have it in our mouths. In fact, on Monday when we mentioned that HIV test, that home test where you swab your gums, that's actually the antibody that you're swabbing for. It's called IgA. But IgA likes to hang out on your mucosal surfaces. So we have it in our mouths, we have it in areas like your reproductive tract. And it's supposed to protect you against foreign invaders. And we're going to talk about antibodies in a couple of lectures, but antibodies are really powerful. Proteins that our immune system cells, they produce. Unfortunately, though, if you are exposed to Neisseria gonorrhoeae, remember we mentioned Neisseria, that was the microaerophile, and this causes gonorrhea. So if you're actually infected with Neisseria gonorrhoeae, one of the reasons why it's so successful is because it makes this enzyme, which basically breaks down the antibodies in your reproductive tract. So the antibodies are basically useless. And Neisseria thrives and does well and the antibodies that are supposed to attack it basically do nothing. So, and we're going to talk about these antibodies. Gonorrhea obviously is an STI. We're supposed to call everything an STI now instead of an STD. But it's one of the most reported sexually transmitted infections. And here at the bottom it says no effective adaptive response. We're going to talk about what that word means, an adaptive. But an adaptive response basically involves your antibodies. So antibodies are supposed to be like the best thing for fighting off a foreign invader. But Neisseria still finds a way to basically sidestep your antibodies, and so a lot of people can acquire an infection anyways, right? And we call all these things STI now is because a lot of the organisms that are sexually transmitted, right? People can acquire them and not necessarily show symptoms, right? A lot of people are asymptomatic. So they might not necessarily show the disease, like STDs. All right, so we went through kind of a good number of examples of extracellular enzymes. The last few things that we're going to talk about. So in the beginning we said there's attachment. Organisms can avoid and sidestep and hide in your body by making extracellular enzymes. And they can also perform something called antigenic variation. In the immune system section, we'll talk about what that word means, antigen, in more detail. But antigenic variation, it's got that word variation. So, kind of sounds like we have a variety of something. It's not just of one type. And we're going to spend more time talking about this in the immunology section. And we'll see that word antigen a lot. So when we see the word antigen, usually we're talking about proteins. They don't necessarily always have to be proteins, but proteins are generally serve as the best kind of antigen. And these are proteins that basically activate your immune system. For sure, these proteins a lot of times, what serve as like the best antigens are the ones that are foreign, the ones that you'd find like on bacteria or viruses. So definitely these proteins can be associated with microbes. And what we're saying is antigenic variation, so kind of one of the themes, definitely in pathogenesis, and HIV actually was a really good example when we talked about HIV. We know that microbes like bacteria, viruses, protozoans can actually alter or change their antigens. Right? We know that, for example, when we talked about HIV was a really good example of this. When we did the whole HIV discussion, one of the things that we kind of emphasized was that one HIV virus is not similar to any other HIV virus, that they're all subtly different. When we say they're all subtly different, the reason why they're all kind of different is because usually the proteins or the spikes or the capsid, they're all just a little bit different from each other. And the reason why they're all sort of kind of different from each other, one of the big reasons why is because we said that reverse transcriptase enzyme is so sloppy, right? And it makes so many mistakes that HIV can definitely alter itself. And so those tiny, and it doesn't have to be a big change. It could be just a minor change. But even if it's a minor change, what ends up happening is, since one HIV virus is slightly different from the next, to your immune system sees it as like totally different. So your immune system is always a step behind. It's always a step behind because your immune system isn't able to like keep up. It's not able to keep up because your immune system reacts against one type of HIV and it's like, all right, I got this. But since there's all these varieties, we're saying varieties, variation, your immune system just cannot find a way to attack every single type of variation. It'll try though. It'll try. So it'll try to attack every single variant, we call them variants, but it won't be able to keep up, right? So I said in a patient who has full-blown AIDS, they say that somebody will typically have billions of different HIV virus within their bodies. So that's kind of a lot for your immune system to take on. It'll do the best that it can. It will make antibodies to as many variations as it can, but it won't be able to win that battle. So we're saying the antibodies won't be able to recognize all those variations, and this happens a lot. HIV is kind of a good example because we just talked about it. We just talked about it, but it shows up, it definitely shows up in a lot of different microbes. So HIV was a good example of antigenic variation. Another good example is this protozoan. So at the top, the name says trypanosoma. This is a protozoan. It's a protozoan, so it's a eukaryotic. It's in that same category as malaria. Remember we talked about malaria, that plasmodium. Trypanosoma is a protozoan that also is like notorious for altering itself. It's got all these variations, like versions of itself. And in fact, it says it has over 1,000 different proteins at any given time. So they're always kind of changing. One protozoan might have a set of proteins, and then another protozoan will have a different set of proteins on the surface. Another protozoan will have a different set of proteins. So your immune system is seeing like all these variations, and it tries to relearn each microbe as it's fighting off all of these organisms. But usually it just can't keep up with all these variations. So, but this is a protozoan. With malaria, we said that was transmitted through through the mosquito, right? Trypanosoma brucei, though, is transmitted through a fly called the tsetse fly. So the fly actually bites you and injects you with Trypanosoma brucei. And the reason why this one's so bad, so your immune system tries to attack it, but it's got all these variations and its antigens. So it totally thrives in your blood without any problem. Eventually it crosses your blood-brain barrier, and it actually infects your brain and spinal cord. And so that's why it's a sleeping sickness, because usually people slip into a coma and it ends up being fatal at that point. So that's another antigenic variation example. All right, and then the last theme for this section of chapter 13 is penetration. Penetration is also another nice tool that microbes use, again, to kind of hide out in your tissues. They can definitely penetrate. So those extracellular enzymes help with penetration, definitely. But another thing that they'll do is they can also release little proteins that allow them to kind of like slip through and sneak into your tissues. We call these invasins, which kind of sounds like invasion. They'll invade. And we do know that some bacteria that are linked to GI infections, like E. coli and salmonella. We're going to talk about salmonella in lecture. That those are like notorious. Once they get down to your GI tract, they'll make these little invasions, and they'll literally kind of like slip through and sink into the cells lining your GI tract. So that's how they kind of literally invade your body, is by using these invasants. And there's kind of like a lot of research focused on these invasants, because people are always trying to figure out how, like, can we use them to make a vaccine or find some sort of preventative? Because salmonella and E. coli, these are two huge foodborne pathogens. They make a lot of people, millions of people sick every year. All right, and I actually have a picture of the gram-negative rod, like sinking in, using its invasants to kind of sink into the lining of your GI tract. So over here on the right, a lot of the gram-negative rods. On the top, I have salmonella doing it. On the bottom, this is actually Shigella, kind of doing the same thing, using the invasants. And salmonella is notorious for doing this. So if you accidentally eat salmonella, it cruises down to the lining of your GI tract, starts injecting invasins into your epithelial cells. And when your epithelial cells get bombarded by these invasins, what they'll do is they'll literally kind of form like a little cup hold around salmonella, and it'll just let salmonella kind of sink in. And that's how salmonella literally invades the lining of your GI tract. So it says it causes the rearrangement of actin is just a protein you find in your cells. So the cells will kind of rearrange themselves, grab salmonella, and literally let it sink in. So it triggers our bodies to take up the bacteria by pinocytosis. So that whole bacteria just literally gets engulfed by your body. So it's kind of a sneaky, creative way that bacteria use to literally invade you. So it's kind of an interesting field of study. And it doesn't really show it in the picture, but salmonella, even as it injects you with invasins, it actually forms like a little needle, and it kind of pokes you and pumps you up with the invasins. And that's what basically tells your cell, all right, it's time to take up salmonella. So that's kind of one of the most important first steps in the infection for salmonella and E. coli and even Shigella. All right. So what we're going to do is we're going to move on and kind of finish up our discussion with toxins, and that'll be it for chapter 13. So we're still on 13. Still on 13. There we go. But our focus is a little bit different because we're going to focus on the damage part. The first part of 13 was more about invasion and kind of like avoiding your immune system. But the second part is more like the damage. So we're going to focus on the toxin production. Alright. And we know that, so with the toxin, one of the things that we'll emphasize is there's no rule about like gram-negatives or gram-positives making more toxins than the other. It falls across both groups. So gram-positives can make toxins, gram-negatives can make toxins. What we're going to do is just do a quick overview of what are toxins. Like when we use that word toxin, what exactly are we talking about? And I notice even now, like in 2016, we use that word toxin a lot when people say, I fasted, so the toxins get flushed out, all these things. But in the context here, like toxins, we're going to talk about toxins that are actually made by bacteria. Bacteria and infectious living organisms. So loosely, we're going to use that word to describe poisonous substances. organisms. So I always get confused. People are like, oh, I fasted for a week to get the toxins out. And I'm like, what are you talking about? What toxins? I'm like, I like to eat, so I think I'll choose to eat. But the toxins, so toxins are real things, but really kind of the focus though for us are, when we use that word, we're talking about actual substances that are made by bacteria or protozoans or worms or whatever it is. In this lecture though, it's mostly going to be from bacteria, and usually toxins are protein based, so they're made up of proteins, and they're literally responsible for making us sick. So it's really the toxins that are like the primary reason why you have the symptoms that you do. Do the bacteria themselves contribute? Of course. So the bacteria are there and they're multiplying, but what's actually causing your symptoms usually is the toxin, as the toxin gets released. Do all bacteria make toxins? No, not necessarily, but a lot of them do. A lot of them will make some sort of toxin at one point or another. The other thing too is that they are super potent. We talked about potency on Monday. We used the terms ID50 and LD50 to kind of get an idea of how potent a bacteria is. On the board, we used anthrax as an example. You can also do potency tests, like instead of endospores, like our example, you can do potency tests on toxins too. So you could do like a similar experiment with your mice or your rats or whatever, and instead of feeding them endospores, you can feed them just toxin and get an idea of how potent the toxin is. And usually toxins are super potent. In fact, we do know that as far as something that's naturally made, not something that's chemically made artificially, but something that's naturally made, we know these are the most dangerous things that we know of as far as naturally made. And they can cause a condition called toxemia. In fact, when we watched the YouTube video, it was like the anthrax YouTube video. They used that word, toxemia. Toxemia means blood poisoning. But the blood poisoning is due to a toxin. So it's got the toxemia. So these toxins, what makes them really bad is they can travel through your blood. So let's say you get sick with E. coli and it's sitting in your gut. You have all this diarrhea. The toxin that's hanging out in your gut doesn't necessarily have to stay in your gut. It can become blood-borne and it can travel throughout your body. So it doesn't have to stay. And we call that blood poisoning. All right, so we know that toxins are super potent. They can cause a lot of damage. And sometimes we have found a way of using these toxins as a vaccine to help protect people. So that's good. That would be like the ideal situation. We take a negative, the toxin, and we flip it and turn it into a positive and we use it as a vaccine. That would be the kind of the ideal situation. All right. I don't expect you guys to memorize this chart by any means. I just kind of highlighted with a blue box. Like some diseases that we might have heard of that are all due to a toxin production. So tetanus is on the list. Tetanus is caused by a toxin. Botulism is something we probably are not familiar with, but that's another disease that we're going to talk about that's also caused by a toxin. Diphtheria, we're going to mention a little bit. That's also, it's a respiratory disease, so it gets moved around through coughing and sneezing. That's also through a toxin. In lab, we mentioned this, but we're going to talk about it also later. Scalded skin syndrome. This is usually something you find in babies when they get a staph infection, and that's all due to a toxin. That cholera, actually, they were just talking about this again with Haiti. whenever there's a natural disaster, usually you have cholera outbreaks. People don't have access to clean water, and that's all mediated by toxin too. And then diarrhea, food poisoning, a lot of that's all due to a toxin. And we're going to talk about the bottom one, which is toxic shock syndrome. That's usually also driven by toxins. So these are a lot of diseases that we've probably heard of some of them, and they're usually all linked to some sort of toxin. So let's highlight like the two big categories. What's good though is we've already talked about one of the categories, so we don't really have to talk about that one too much. It's really the first category that we're gonna focus on. So toxins are subdivided into two groups. You're either called an exotoxin or an endotoxin. But you guys already know what endotoxin is. Endotoxin is found in what molecule? The endotoxin. Yeah, that was the LPS, right? So we're going to focus on the exotoxin, the first category, because that's all brand new to us. But you guys have seen that prefix exo before. Whenever we see exo, what does that mean? Outside. It's outside. Like even with malaria, we talked about the exo or erythrocytic stage, right? So that's all happening outside. Same thing here. So these are toxins that they're produced inside, but their final destination will be outside. So the final destination is that toxin will actually be released out of the bacteria. The bacteria secrete it out, and so that's why it says produced inside and then secreted outside by the bacteria. And we did say though that really the majority of exotoxins are all protein based. So they're really made up of proteins. And we also said there's not really a rule about like, oh, only gram negatives can do this. It actually crosses both categories. So gram positives can make exotoxins too. Now, in order to make an exotoxin though, you have to have the blueprint, the information to build it. And that blueprint you usually find in two places. Definitely one of the main places you can find the blueprint are on those plasmids, right? So it's usually the plasmids that carry the blueprint for toxins. If bacteria don't have the blueprint at all, they probably can't make the toxin. They cannot make the toxin. It has to have the blueprint somewhere. Now, unfortunately, though, in the beginning of the semester, we said those plasmids can get shared through things like conjugation. So if a bacteria were to like pick up a plasmid that it never had before, it could acquire the ability to make a toxin when before it was totally fine, it wasn't making a toxin. So that can happen. And then the other thing is those viruses, remember we call them bacteriophage, and they have like those little legs, and they go around and they squat and they infect the bacteria. Those viruses also can carry the blueprint for toxins. So if you're a bacteria and you accidentally get infected by a virus, you can also pick up the blueprint that way. So either way, those are the two most popular methods for acquiring the blueprint. Now, endotoxin, we're not really going to talk about that much because you guys are kind of already familiar a little bit. Remember we said endotoxin, now that's kind of a different subcategory. Endotoxin is that LPS, that lipid part of LPS though. So this is a different category because of course endotoxins are only found in gram negative. Because gram negatives are the only ones that have the LPS. Gram positives don't have LPS. So that's why it says only associated with gram dumped out in your body. So that's good. At least it's not as potent. So our focus is going to be in the top category with the exotoxin. What we're going to do is talk about the subcategories of exotoxins. Here are my 3 big subcategories. All right. So in the exotoxin category, we're going to focus on these 3 groups, these 3 subcategories. The top one says AB toxins, and then the middle one says membrane disrupting, and the third, basically big subcategory, is called super antigens. These are the 3 most common subgroups within the exotoxin. So remember, pretty much everything is protein-based. Can be made like gram-positives, gram-negatives. They're all 3 released. Bacteria actually dump them out in your body. They can travel around for sure, so they don't necessarily have to stay put right where the bacteria is. And definitely some of these, we've turned them into vaccines. It's mostly been the AB toxins that we found a way to put in a vaccine. So we're going to start with the AB toxin category first. So just to give a couple examples of the AB. So within that AB category, usually there's kind of like different examples of where the toxins like to have their effect in your body. So one example is a category called neurotoxins. So if we call something a neurotoxin, what we're basically saying is that this is an AB toxin that has its effect on your nervous system. So we're gonna delve into some more specific examples of neurotoxins later on in the semester, and how they wreak havoc on your neurons in your nervous system. A lot of times they cause a lot of damage. So tetanus is a good example, and botulism is another example. So botulinum toxin falls in this category. And you guys already told me before, we found a way to commercialize botulinum. How is it sold to people now? Botox, yeah. So that's basically what Botox is. So when people get their Botox little injections, it's actually made up of botulinum toxin. So you're actually getting toxin injected into your body. And they didn't like change it or modify it. It's the straight toxin that they took out from the bacteria. And if you think about this, is like a genius idea. Whoever thought of this is like genius, because the company that makes Botox, which is in Orange County, which I think is very fitting, they're actually based in Orange County, and that's like the #1 way that they make their money is Botox. They do other things too. We're going to talk about the company. They do other things, but Botox is the #1 money maker. But it's genius because for them, it's probably hardly 0 to nilch money as far as for them to make it, because it's just coming from bacteria. But the markup, right, it's pretty expensive. And just like with a lot of the other products, you need to kind of continually keep up with it. So you can't just get like your one Botox shot and be done for like a year or 2. You have to keep going, getting more and more and more. So as far as like, you know, money maker, it's genius. It's a genius idea. So neurotoxins. The other kind of subcategory are enterotoxins. And whenever we see that prefix entero, that means GI tract. So these are toxins that have their effect in the lining of your GI tract. So they definitely cause intestinal symptoms. And there's a lot of enterotoxins. So unfortunately, there's many ways that we can get food poisoning. And enterotoxins is is definitely a big reason why. That cholera that we talked about that causes that profuse, watery diarrhea, that's all driven by a toxin. So the toxin is what stimulates your GI tract to dump out water. Our GI tract is supposed to absorb our nutrients, but cholera toxin actually tells your intestinal cells to do the exact opposite. So it tells your cells to start dumping out water, and that's why you're losing the liters of water a day. And then my last category is this. It's a cytotoxin. And a cytotoxin, what this subcategory does is it basically has a pretty fast killing effect on your cells. So it lyses them, which is bad. Or it finds a way to shut something down in your cell, and then your cell kind of auto-destructs and dies. So what I'm going to do is kind of use diphtheria as a good example. We're going to do like just a very brief overview of how diphtheria does that. So these are kind of the three big subcategories. Later on in the disease section, we're going to talk about a lot of these toxins in relation to different diseases. So let's mention the diphtheria. That'll be our good example. What I did not tell you yet though is what is the A, B? What does that mean exactly? The A and the B. So the A part and the B part, those are letters for designating the two different parts of the toxin. We call it an A-B toxin, and the letters are used to kind of designate which part does the binding, that's the B. So we use the letter B for binding. And the A, you can kind of think of it as the effect that the toxin has. So the A is the part that does the damage. The B is the part that just binds. Every toxin, it has to bind. If you are an AB toxin, the first thing that the toxin will do is that it will find some place in your body to bind, to attach somewhere. If it's an enterotoxin, it'll bind to your intestines. If it's a neurotoxin, it'll bind to a neuron, somewhere in your nervous system. If it's in that cytotoxin category, though, a lot of times, unfortunately, the AB can kind of bind wherever. It may not necessarily have a specific niche, but it will bind, and then eventually it'll have an effect on your cell. So let's use diphtheria as a good example, because it's kind of a good little picture of what diphtheria does to us. Now we're going to mention a little bit about diphtheria, because diphtheria, it's a respiratory disease, so it moves around through mucus, coughing, and sneezing. And usually kids will get vaccinated. It's included as a part of the childhood vaccination series. But the reason why diphtheria is so bad is because, let's say somebody coughs or sneezes on you, so you get exposed to the bacteria. That bacteria can actually dump out an AB toxin in your body, and the AB toxin can actually get released into your upper airway. So this is a bacteria that likes to hang out primarily kind of the very back of your throat. And sitting there, it can dump out its AB toxin. The toxin, though, is not a neurotoxin or enterotoxin, it's a cytotoxin. So we just said cytotoxins can lyse or alter the function of your cells somehow. So what happens is the B part will go ahead and bind to the cells at the back of your throat. After it binds, the whole toxin will actually enter your cells, the whole entire thing. And then eventually, what the A part will do, because we said the A is what does like the damage, it has the effect. What the A will do is it'll start to shut down your ribosomes. So we definitely have ribosomes in our cells, and you guys already know ribosomes are the place where we're building protein, right? So if that toxin starts shutting down your ribosomes, your cells are just going to auto-destruct and die pretty fast, because there's no way they can survive without their ribosomes working properly. So those cells start dying, and then what's really bad is if your infection progresses, if it continues on and you don't get any treatment, that toxin, not only is it being made in your pharynx, in the back of your throat, it can actually start to spread. So the toxin can get in your bloodstream, spread to other areas. Heart is obviously a critical one. So for children, this can actually be very deadly, especially kids under 5. It can cause heart damage, shut down kidneys, and shut down the lungs. So it can start shutting down ribosomes throughout your body. Doesn't necessarily have to stay in your airway. So I have a picture because diphtheria in kids, like one of the first symptoms that develops is it's called like a bull neck. So the lymph nodes swell up really, really big, and you start to attack the bacteria. And then we said that the bacteria primarily takes up residence at the back of your throat. As the toxin gets dumped out, that kind of slimy, mucousy layer, what ends up happening is we said that the toxin disrupts your ribosome, so your cells start auto-destructing. And when they start dying off, what ends up happening is as those cells die, you end up building up like this thick tissue. And it can get so thick, you can see it on this side and that side, they're like already pretty wide. That's all dead cells, And it can build up to the point where you have so much damage and it thickens up so much that you can actually suffocate. It'll cut off your airways. So that's why the protection through the diphtheria vaccine is so nice. And it's actually included, we'll talk about this later on, it's in that DTaP shot, right? So it's included with your tetanus shot a lot of times. It's a combination. So that's one example of how AD toxins work. All right. So the middle category is the membrane disrupting toxins. What we'll do is we'll mention the 3 categories and then I'll show you guys a video, kind of summarizes. Membrane disrupting toxins. So in that other subcategory, it's not just all about AD toxins. These are toxins that damage membranes, cell membranes. And unfortunately, every single cell in our body has a membrane associated with it. So as these toxins get dumped out, usually they'll end up lysing your cells somehow. So it says one way this can work is if a toxin gets dumped out, it can actually disrupt your membranes, causing them to lyse. And the way that they do that is they poke little holes in your membrane. So they form like these little pores, and they poke holes. Wherever there's a membrane in your body, they'll start doing that. And the 2 most popular membrane disrupting toxins, we call them hemolysins. It's got the word lice in it. It kind of ends in the lice. Because these are toxins that lice your cells. So again, here's that Staphylococcus aureus. That's the bad one. It makes a hemolysin that literally forms pores in your cells and causes them to lyse open. Now we're going to talk about the top one in lab actually. Good. So you guys did your throat cultures. Basically, the way that we're going to know whether you're carrying the strep throat organism, and the organism that causes strep throat is actually this one, Strep pyogenes. The way that we're going to know if you're a carrier is we're going to look on the plate and see if the bacteria that you picked up on your swab, if it's dumping out a membrane-disrupting toxin, that's going to lyse the cells on your blood agar plate. So we're actually going to look for, tomorrow in lab, the membrane-disrupting toxin here, called streptolysin O. That's what we're going to look for. So, and if that toxin is being dumped down on your plate, then we'll know, all right, you're probably a carrier. So we'll earmark you. Yeah, you will kind of get a little bit of a halo. Mm-hmm. Yes. So pore formation is also popular. And then the other one is, it says phospholipases. What phospholipases do, it's just a little bit different. Instead of like forming a pore that kind of punctures, it's almost like a puncturing effect. Phospholipase will just cause kind of like chaos in your membrane. So that phospholipid bilayer is arranged all perfectly with the hydrophobic part outside and the hydrophilic component on the inside. But these toxins causes just total disruption and disarray of your bilayer. And so your cells also lyse. And they basically burst open and die. And one organism that's like notorious for doing this, it says Clostridium perfringens. Now you guys already know this from the previous chapter. What's so unique about the genus Clostridium? Yeah, endospores. So Clostridium is one of those organisms that's just kind of, it's all around us, and it just hangs out as an endospore. Unfortunately, though, if you have some sort of like major trauma or wound, or you're just really susceptible, you're in that diabetic category, if you get exposed to Clostridium perfringens endospores, and the endospores are just kind of all around, they're environmental. Once they enter your system, they can also reawaken, we said germinate, start releasing membrane damaging toxins, and those toxins can actually cause gas gangrene. When we see that word gangrene, what's happening to your poor cells? They're dying, right? It's kind of like massive death of your tissues. Gangrene can definitely be life-threatening. You can develop gangrene from different reasons, but a bacterial infection is definitely one reason that can contribute to gangrene. So as this toxin, now if you don't get this infection under control, that toxin can continue to spread and you just get more and more and more cellular tissue death. What makes this type of gangrene a little bit different is the gas kind of helps you as a clue. The gas is always a helpful clue that it's probably bacterial. Because what happens is as the tissues are like dying off from this bacterial infection, usually there's gas that's made as it's fermenting the sugars in your body. So as it's fermenting, it starts becoming bubbly. Your tissues can become a little bit bubbly. And that's the gas from fermentation. All right, so I actually have a picture. This is what you guys are going to look at tomorrow. The blood agar. The blood agar plate. And you guys streaked it out, just like in this picture. We did the 4 quadrants, and hopefully, just like in this picture, like in the 4th quadrant, we'll start to see little individual colonies because you basically like diluted out what was in your throat, and we made it less and less and less less concentrated. And then what we're going to look for is that toxin production, that membrane disrupting toxin that'll actually damage the red blood cells in the blood. This is all blood. And it's full of red blood cells, so if you're positive, we'll be able to see that toxin effect, and it'll basically kind of look like it's glowing a little bit, right? So we'll look for that. For the gas gangrene, we said that's basically like massive tissue death, and that's all due to bacterial infection from Clostridium, which it's hard to avoid because the endospores are just kind of everywhere. But once it enters your body, as it dumps out its membrane disrupting toxin, that toxin totally results in the lysis of your cells, and so your cells literally start dying off. And that can spread and spread and spread. So we have one last category, and then I'll show you guys a little animation. My last category is this, the super antigens. Now, at least on the board, we wrote out what that antigen word meant. We said it's usually proteins that like stimulate your immune system, and that definitely microorganisms are coated in antigens. They're coated in proteins that stimulate your immune system, right? That kind of get it going and activate it. The only bad thing is it's got the prefix super. So these are like really, really, really potent antigens. We call them super antigens. And these are exotoxins that they like to bind to your T cells. So this is a type of toxin that seeks out a T cell. A T cell is a type of white blood cell, right? That we have in our blood. And what we're going to do is draw this out, what that means to trigger massive cytokine release. So we'll sketch that out. But they'll bind to your T cells and they'll get your T cells activated. Now that doesn't sound like such a bad thing, but the problem is your T cells, they basically kind of get overstimulated. So it says mass activation. It's always good if we get our white blood cells activated and fighting against something, but it's kind of a balancing act. Because if our white blood cells get overactivated, they get overstimulated, your immune system can actually work against you and start causing damage to your own body. And that happens a lot. So this is what happens with the super antigens. Your immune system gets overstimulated, and what we're going to draw out on the board is this is what happens with toxic shock syndrome. Toxic shock. Right? So they always give you the warning about toxic shock syndrome on the boxes of tampons, right? It's not as common today as it was when they were first manufacturing tampons, but like Jalen, it still happens. We found an article, there are recent stories where women still get toxic shock even today, just from their tampon usage. So that's what we're going to sketch out as our example. Okay. So it's not as common, but it still happens. The article that Jalen had found, it was a young girl in her 20s who got toxic shock. And we'll sketch out the toxic shock. There's a lot of different ways people can go into shock. Definitely bacteria is one of them that contributes. In my example, we're going to talk about staph, but it can be a variety of bacteria that cause you to go into shock. So I'm just going to use one example, but definitely another example is we did mention gram negatives can cause somebody to go into shock too, not just staph. Because gram negatives have all that LPS, right, that gets released out. But in my example, though, I'm going to talk about the super antigen example. And if she's using tampons. And what we're going to do is this is all happening in the reproductive tract. And usually there used to be a really high rate of tampons. In the past, the problem was with the super absorbent tampons, because originally when tampons were created, they wanted to make them as convenient as possible, so they kind of made these mega ones, so that women wouldn't have to change them very often. It actually ended up being a bad thing. So now they've gotten rid of super absorbent tampons. But the problem with tampons, we know tampons collect blood, and that blood can serve as a nice nutrient-rich environment for bacteria to collect in, right? And one of the bacteria that actually does really well in that blood-rich environment is Staph aureus. Now, Staph aureus, I would say, like, the usual kind of niche for Staph aureus is not like the vaginal tract of women. So it would have to be introduced into your vaginal tract somehow. We know there's all sorts of ways that can happen. For sure, your hands. So have clean hands before you insert a tampon. We carry Staph aureus in our hands all the time. But yeah, the bacteria would have to get introduced into the reproductive tract. But once it's in there, if you have blood that's collecting, now the story that Jalen and I found, the young woman who got her toxic shock, she actually did not have the tampon in there for very long, so it wasn't like a length of time thing. But Staph aureus, since you guys know, is a grape-like cluster. It's perfect because it has all that blood with hemoglobin in it. It's got plenty of food, and it can actually start to multiply in the vaginal tract. As it's growing by binary fission, eventually it can start to dump out toxin. And the toxin that Staph aureus likes to dump out is a little protein called a super antigen. Now, unfortunately, what's really bad about the super antigens is we said they're usually all little protein-based, and they're really, really small. And they're usually small enough where eventually these little proteins will get absorbed into your blood. So they're very tiny, they'll start traveling around. And the super antigens, the type of host cell that it loves to bind to actually is your T cells. So it'll start looking for T cells. A T cell is a type of white blood cell that you find in your blood. And we're going to talk about this in the immunology section later on, but T cells are coated with little receptors. Like one of them is CD4. We talked about that with HIV. These little receptors. And the receptors are kind of like little homing beacons. And what they're doing is they're just waiting to attack like anything that's foreign or see anything that's foreign. Now the super antigens, what they do is they kind of cruise along and they latch on to these little receptors. And they kind of latch onto them. And unfortunately, once your T cell kind of gets that signal, it becomes very, very stimulated. And your T cells will become activated. So they become activated. Whenever you get a white blood cell, when we say becomes activated, usually it's always two things that happens. One is now your T cell, this T cell will start dividing like crazy. So they'll start multiplying. You'll get like this big old pool of T cells. Start dividing. Dividing T cells. So that's one thing that happens. But the other thing that happens is after they're done dividing like crazy, is usually they'll start doing whatever it is that a T cell is supposed to do. So they'll start reacting. They'll divide and then they'll have their reaction. One of the reactions though of the T cells is the T cells, one of the things we said is they like to help out and they kind of coordinate everybody else. So these T cells will dump out chemical signals, and it's basically the chemical signal that makes you sick. So kind of the sad part of this is it starts off with bacteria making a toxin, but in the end, what actually ends up causing all the damage in your body is the chemical signals from your own white blood cells. That's what actually makes you sick. So those T cells divide like crazy, but then the second part of this is your T cells release chemical signals. And the chemical signals that get produced, there's a name for them. They're called cytokines. Cytokines are the chemical signals that T cells like to dump out. This is how they talk to each other. This is like their form of communication. Your immune system can communicate with each other. They talk to each other all the time. And it's through chemical messages. Kind of the same idea like how our nervous system has neurotransmitters. Like that's how they communicate. In the world of your immune system, it's through these little messages called cytokines. So it's kind of the same idea actually as neurotransmitters. So your T cells go crazy. They got stimulated by this super antigen. The super antigen is telling them, all right, divide, start dumping out your chemicals. And the chemical that it dumps out is called TNF. Remember whenever we see letters, this is a protein. And it's a chemical message.
Professor?
Yes.
So this occurs because the super antigen latches onto the receptors, right?
Yes, yes. Yeah, so the super antigen latches onto your T cell, and that's what kind of tells them to become so stimulated. And this is how they're becoming stimulated, so they start dumping out their chemical message. And unfortunately, this particular chemical message tells your capillaries to dilate. Why would that be so bad? To have all of your capillaries dilating and becoming leaky. Why would that be such a bad thing?
They'll drop your blood pressure.
Yeah. So dilation means they're in your capillaries, you're going to start to get very leaky blood vessels. your blood plasma is going to actually start leaking out of your blood vessels. They're becoming very dilated. Now, a little bit of dilation is okay. That's actually a good thing. Like when you get sick with something or you get trauma, a little bit of dilation is okay. It brings more blood flow. Now it's not comfortable. You get very inflamed. You get inflammation, redness, heat, all these things. But if it's a lot of dilation, which is what's happening here, because your T cells are going crazy and they're going overstimulated, all this dilation will cause you to go into shock. Which is basically what happened with this young woman. And she almost died from her infection. So she went in to see her doctor originally, and they didn't really understand what was going on with her. By the time she finally was admitted, when you're in that state of shock and your blood pressure is totally plummeted, and obviously you're very disoriented, you're really confused, and all of those things happen in that particular case. And that's all due to the start of our Staph aureus infection. But we did say gram-negatives, unfortunately, can also cause shock, not through your T cells, but through that endotoxin. So they can also have a shock effect. So that's the super antigens. Yeah, we said that they like to latch onto your T cells. That's like the primary place where it gets all started. All right, let me switch to my little animation. Hopefully this cooperates.
So what exactly makes LPS endotoxin? An endotoxin? Because it still secretes out the toxin, right?
Oh, they're just saying it's endo because but it's not. It's only released out. Like the bacteria don't intentionally release it. Only gets released if the bacteria dies.
Okay.
Yeah. Like exotoxins, the bacteria actually secretes it.
Right.
Endotoxin, though, it's not secreted. It just sits in the outer membrane. LPS.
Yeah.
So the only way it would get dumped out is if the bacteria died, and it kind of destructed, and then that's why it got released.
Like if a white blood cell
Attacked it?
and kills
Yeah.
it. Okay.
That would be one way.
Alright.
Yeah. So, so that's a good question. Like for endotoxin, because we mentioned endotoxin just sits in the outer membrane of gram-negatives. So the bacteria is actually not actively releasing it. It just kind of sits there. So it's only a problem if that gram-negative were to die somehow. Like your immune system attacks it, and then that's why the endotoxin gets released. It's not like these exotoxins that are just kind of perpetually getting dumped out by the bacteria. So we'll see that in this. Okay, let me up my volume.
Bacterial proteins that are secreted by a living bacterium into its surroundings. In a host, these exotoxins circulate and cause damage to host cells in various ways. Exotoxins can cause severe damage or death at very low concentrations. Exotoxins can be classified in different ways. One method classifies the exotoxins by the area of the host that is infected. For instance, neurotoxins are toxins that affect the function of neurons. Tetanus and botulism are caused by species of Clostridium that secrete neurotoxins. Enterotoxins, like those secreted by the cholera bacterium, affect the lining of the intestinal tract. The term cytotoxin refers to an exotoxin that kills or seriously damages host cells in general. Corynebacterium diphtheriae is a bacterium that secretes a cytotoxin that inhibits protein synthesis in host cells. Exotoxins can also be classified by their structure and function. This classification scheme has three categories: A-B toxins, toxins that disrupt membranes, and superantigens. Most exotoxins are proteins with two domains, A and B. The A domain of the exotoxin is an enzyme that has a particular function. The B domain of the exotoxin is the binding component. It binds to a specific receptor, usually a carbohydrate on the host cell. Once the B domain attaches to the host cell uses endocytosis to bring the exotoxin inside. Then, the B domain opens a pore in the endosome, allowing the A domain to escape into the cytosol, where it can inhibit protein synthesis or interfere with the host cytoskeleton. This usually results in death of the host cell. Membrane-disrupting toxins exert their effect without entering the cytosol. They can form protein pores in the host plasma membrane. Or they can disrupt the phospholipid portion of the membrane. In either case, the cell lyses. Superantigens are bacterial exotoxins that stimulate an excessive immune response. First, it causes a proliferation of T cells. Then it causes the T cells to release excessive amounts of cytokines, which can stimulate fever, inflammation, and shock. This very intense response could lead to death. The toxins secreted by Staphylococcus aureus in food poisoning and toxic shock syndrome is an example of a superantigen.
All right. Okay, so the last thing that we're going to mention is actually kind of related to Sim's question about endotoxin. That'll be it for today. So endotoxin can also have a shock-like effect. Not because it's dumping out superantigen into your bloodstream, but because that LPS is accidentally getting released somehow. Now sometimes that can accidentally happen, usually when the bacteria dies for some reason. These are the three primary reasons why that bacteria would die and dump out LPS. Sometimes it just happens as a part of the binary fission process. Usually though, unfortunately, your own immune system is like attacking the gram-negative. That happens all the time, right? So our white blood cells are phagocytosing, they're eating the gram-negatives. And in that phagocytosis eating process, LPS gets dumped out. And then sometimes this even happens with antibiotics. When people initially start taking their antibiotics, they might have a little bit of a fever before the gram-negative is cleared out. So if a little bit of endotoxin gets released out, a lot of times people will experience just a fever. If it's a lot of endotoxin that gets released out, it could result in that shock-like effect. So the last thing that we're just going to kind of sketch out is how that endotoxin can actually trigger a fever response. A mild fever is generally considered okay, right? But what's so dangerous about a really high fever? We start talking about 102 and up. Yeah, so that high body temperature, right, can unfortunately cause damage to the proteins in your own body. So really high fever can potentially be a little bit more dangerous, right?